Hello friends, welcome to EPG Pathshala. I am Dr. Anita Kamra Verma, Department of Zoology, Kirodimal College, University of Delhi, and I would be teaching you leukocyte activation and migration. This is module 19 of the immunological series. At the end of this lecture, the students would be able to appreciate the migration of cells, which is a critical component of both innate and adaptive immunity, as the immune responses heavily rely on the ability of the leukocytes to move from the circulation into the tissues. The foremost aim of this lecture is to discuss the mechanisms and consequences of neutrophil recruitment, lymphocyte recirculation, and monocyte trafficking, all of which require adhesion, transmigration through stimulated blood vessel walls. The mechanisms that mediate the initial phase of leukocyte endothelial cell contacts within the lumen of the blood vessels will be discussed, in particular the migration through the perivascular basement membrane, which is a barrier which is rarely investigated in the context of the immigration in the in vivo system. So, how do we define leukocyte trafficking? It is just the movement of the cells from the blood vessels into the tissues, and leukocyte trafficking can be divided into two major phases. One, movement within the vascular and the lymphatic circulation, and migration within the tissues. The vascular and the lymphatic circulatory systems are lined by monolayers of endothelial cells that grow on an abluminal layer of extracellular matrix, the basement membrane. These cells form organized intercellular junctional zones that include adherence junctions, tight junctions, and gap junctions. Hence, the endothelium serves as the principal, that is the selectively permeable barrier between the circulation and the underlying tissues. Therefore, each phase change during trafficking, that is movement into the circulation, which is termed as intravasation, or out of the blood vessels, that is extravasation. The circulation therefore requires that the leukocytes cross the endothelium, a term called as diapodices. Now the fundamental question is, why do leukocytes migrate to the tissues? Leukocytes are derived from the bone marrow progenitor cells and found in circulation as blood monocytes and differentiate as myeloid dendritic cells into the periphery and into the tissues as macrophages. Cells of the immune system constantly move throughout the body to fulfill their role of immune surveillance and pathogen elimination, such as blood leukocytes, which include lymphocytes, monocytes, dendritic cells, and neutrophils, all of which cross the blood barrier and travel to the site of infection. The phagocytic activity of leukocytes, coupled with their ability to segregate into antigen presenting cells, contributes to the innate and adaptive immune response. The threat signals caused by the inflammatory cells or the inflammation and injury stimulate the innate arm of the immune system that consists of macrophages, mast cells, and dendritic cells, all of which secrete pro-inflammatory cytokines to activate the blood vessel endothelial cells. A critical component of both innate and adaptive immune system is the migration of leukocytes from the circulation into the tissues. So, we must understand the importance of cell adhesion molecules at this point. For circulating leukocytes to move to the inflamed tissue or the peripheral lymphoid organs, the cells must adhere to and cross the endothelial cells lining the blood vessels. This is the process which I have said is called as the extravasation. The endothelial cells express leukocyte specific cell adhesion molecules, which are commonly called as CAM. CAMs on the leukocytes help to adhere to the vascular endothelial cells and to enhance the strength of the interactions among the cells of the immune systems. That is, T helper cells will combine with antigen presenting cells, or the T helper cells with B cells, or the CTLs with the target cells, CTLs being the cytotoxic T lymphocytes. Now, structurally, CAM can be classified into four families. mucin-like CAM, which expresses sialylated aldehyde side chains or moieties, an integrin family, which is a heterodimer consisting of alpha and beta polypeptides, selectins, wherein the L-selectin is expressed on the leukocyte and P and E selectins are expressed on the inflamed endothelium that expresses CD62, that is cluster of differentiation C62 that ends in a lectin domain. There is an immunoglobulin superfamily CAM that has a fibronectin type of domain that associates with the integrins. Now, I must also explain that there are three types of selectins, the L-selectin, which I said is expressed by the leukocytes, P and E selectin, which are expressed by the endothelium, and there is mucin-like CAMs, that is the glycosylated CAMs, which is expressed on the endothelium, and CD34 on the endothelium, but PSGL-1 is expressed on the neutrophils. That is, mucosal associated CAM1 is also associated and can be combined with the P-selectin. Now, there are different integrins, that is VLA4 selectin, that is a very late antigen 4 type, and there is an LPAM. All these selectins, integrin molecules also combine with P-selectins for the activity or for the adhesion and transmigration of the molecules. Similarly, immunoglobulin superfamily also has intercellular cell adhesion molecules or vascular cell adhesion molecules, which are commonly called as ICAM1 and VCAM1, or LFA2, that is the leukocyte function associated antigen or mucosal associated cell adhesion molecule, which is expressed on the mucosal epithelium. At times, both are mucin-like and immunoglobulin-like domains occur simultaneously. Chemokines facilitate both activation and migration. Chemokines are small peptides made up of 90 to 130 amino acid residues. They have the ability to modulate chemotaxis, adhesion and activation of leukocytes, regulation of leukocyte trafficking. A few of the chemokines are involved in inflammatory responses. Some are constitutively expressed and have an important role in homeostatic or development of the cells. Chemokine mediation or chemokine mediated effects may not be restricted to the immune system only. There are pro-inflammatory chemokines. They can induce a response to infection and they are capable of recruiting phagocytes and lymphocytes to the inflammatory sites. There are 4 classes of chemokines: CXC, CC, C, CXXC. The ligands which are expressed are called as CXCL ligand and the receptor is called as the CXCR receptor. Now, coming to leukocyte activation and migration. The multi-step model of leukocyte transmigration includes tethering to slow rolling and regulated rolling of the leukocytes across the vascular endothelium. A schematic diagram showing the sequential steps involved in leukocyte transmigration through stimulated blood vessels, indicating the capture for the transitory adhesion, rolling, slow rolling, firm adhesion and diabetesis or transmigration across the endothelium is expressed. The adhesion cascade includes capture, arrest, adhesion, strengthening, intraluminal crawling that involves spreading, paracellular and transcellular diapetesis, also migration through the basement membrane. So, under the physiological conditions, the endothelial lining of the vessels present an anti-thrombogenic, anti-adhesive surface to ensure blood flow along with the blood components, thereby maintaining homeostasis. That means, under normal circumstances, there is no question of the blood cells moving out of the blood vessel. The neutrophils will move slowly rather than they roll over the endothelium, unlike the other blood components that move along the blood flow with a high velocity. But at the site of injury, the place of infection or the place of inflammation, the presence of endotoxins Expression of certain cytokines and chemokines and other secreted molecules render adhesive properties to these circulating leukocytes, thereby initiating a cascade of leukocyte immigration. The slow rolling then goes into a response and generally mediate a weak and irrevocable molecular interaction between the selectins, that is, I have already explained the P-selectin, E-selectin and L-selectins which are present on the endothelium, and their counter ligands, the PSGL-1 present on the leukocyte. In addition, certain integrins such as very late antigen 4 can also mediate leukocyte rolling under conditions of blood flow. The slowing down of leukocytes during rolling facilitates the ligation of specific and high affinity G protein coupled receptors by the endothelial cells of the lumen, associated activating factors such as chemokines. This interaction augments the ligand binding profile of integrins, that is achieved by increased affinity and avidity of the molecules. Integrin activation plays a critical role in facilitating the adhesion of leukocytes firmly to the endothelial cells, leading to flattening of the leukocytes over the endothelium, thereby ensuring two crucial steps in leukocyte immigration. Beta 2 and beta 1 family of integrins are the major integrins involved in the step of leukocyte or endothelial cell interaction. Therefore, the interaction of leukocytes with the vascular endothelium is a highly coordinated process that includes the arrest of the free flowing leukocytes in the blood with subsequent leukocyte rolling arrest, firm adhesion and ensuing the diapedesis. These interactions will happen only under high shear stresses within the blood venules and will depend on the multiple families of adhesion molecules as discussed above. As a response to infection, mediators and the leukocyte gathering is considered to be very important for an adequate defense of the host organism to any kind of injury or infection. Activation of endothelium also contributes largely to the systemic inflammatory responses to septicemia and the enhanced expression of an immune response. Within the setup, leukocyte transmigration through vessel walls of the post-capillary venules is the concluding stage of a cascade of responses of leukocyte mediation by a sequence of molecular interactions that primarily mediate the slowing down of the velocity of the leukocytes, rolling that has been followed by firm adhesion of the leukocytes to the endothelium, and finally the transmigration through the vessel walls of the blood vessels. Leukocyte transmigration acts as a means of guiding the emigration of the leukocytes from the lumen to the blood vessels to the extravascular tissues, thereby playing a role in monitoring the phenotype of the immigrated cells in such a way that responsiveness of behavior of leukocytes to chemoattractants directs the migration and interactions with the molecules of the extracellular matrix or the extravascular tissue that may be further regulated. Therefore, such a mechanism clearly contributes to the capability of the immune system to elicit an appropriate, optimal and localized tissue response to a variety of extravascular stimuli without triggering any non-specific damage to the vasculature. Therefore, leukocyte migration requires polarization. Leukocytes that adhere tightly to the vascular endothelium need to breach or break this vascular barrier to reach the underlying inflamed tissue. This can only be achieved by active integrin mediated cell migration. That is, the migration also requires polarization of the cell body and the formation of a lamellipodium. It also requires the formation of a uropod at the tail of the leukocyte. The formation of these structures are driven by integrins and the cytoskeleton. Nevertheless, in a situation of uncontrolled infiltration of leukocytes that may exceed either in duration or in magnitude or is incongruous in its localization, the transmigration induced alteration in the phenotype of the leukocytes may lead to stimulation or exemption of the deleterious inflammatory responses in the host. Transendothelial migration of leukocytes. Formally, the basic steps for intravasation should include interstitial migration towards the vessel, migration across the basement membrane, and across the endothelial barrier. Again, it is diapedesis. And ultimately, the release of the luminal leukocytes into the circulation. It is now appreciated that diapedesis, whether it occurs during intravasation or during extravasation, will occur in two distinct routes or pathways, either via the disassembly of the intercellular junction to form a paracellular gap, or via the formation of a transcellular pore directly through the individual endothelial cell, that is the transcellular diapedesis. The concept of one cell passing through or in effect entering another cell, as occurs in the transcellular diapedesis, might seem very bizarre and a very unlikely event. When integrins are activated and the cell migration machinery is functionally polarized, the leukocyte traverses along the luminal side of the vascular endothelial cells and then transmigrates primarily by transiting between the neighboring endothelial cells. Nevertheless, leukocytes have the ability to pass the body of the endothelial cells too. Although there is a prevailing transcytotic pathway for extravasation, it may not be the crucial route which is often taken by the leukocytes. A leukocyte taking the conventional paracellular route faces the vascular cells connected by a large array of endothelial junctions that require to be penetrated. These junctions preserve the integrity of the endothelium and monitor the vascular permeability. Three types of endothelial junctions have been described. Just to revise: tight junctions, adherent junctions, and gap junctions. The endothelial junctions resemble those found in the epithelium, but their three-dimensional organization is often disordered, indicating that the perpetual remodeling that commences from leukocyte trafficking. Undeniably, transmigration is a very vibrant process in which the tight junctional, gap junctional, and adherence proteins facilitate the final step of leukocyte extravasation, functioning as a liquid engulfing and transmigrating cells rather than just functioning as a gate. So, recruitment to non-inflamed tissue is another reason to understand why we want to understand leukocyte migration. When there is a lack of an inflammatory trigger, the adhesion molecules along with the chemokines that are involved in the collection of macrophages and dendritic cells in peripheral tissues should be constitutively present. The dendritic cells and the macrophages originate from the bone marrow and hence are required to be recruited from the circulating precursors in the blood. Amongst the constitutively expressed chemotactic molecules that act on the circulating monocytes are CXC chemokines and CXCL14 ligand, which is again identified in the breast and the kidney, expressed as chemokine BRCA. This has been projected to be constitutively present on the trafficking monocytes. However, the role of CXL14 on other chemoattractants such as defensins has yet not been explored with regards to the recruitment of the monocytes to the non-inflammatory tissues. Now that we have understood the trafficking of the leukocytes, we must understand T lymphocyte recirculation is extremely important to understand, and this also occurs via multi-step pathway. The maximum exploratory behavior among cells is displayed by the T lymphocytes. They are capable of migrating throughout the body and are able to explore most of the tissues. Besides large-scale displacements, T lymphocytes continuously probe their microenvironment in a mode preferring recognition of specific antigens at the surface of the antigen-presenting cells. Mobility is a characteristic property of T lymphocytes that promises timely and optimal recognition of extraneous antigens at the site of the antigenic challenge. When T lymphocytes travel across the venular blood vessel walls to enter various tissues and organs, it is called as transmigration. The ability to migrate is crucial for the T lymphocytes at all the life stages, from entry of the T cells precursors into the thymus for T cell expansion, to entry of the naive T cells into the lymph nodes for activation, migration and proliferation of effector cells into tissues to combat the infections. So, transmigration is also a multi-step cascade that includes lymphocyte rolling, signaling, firm adhesion and transmigration. Each step is constantly regulated by interactions among adhesion molecules and chemokine receptors that are expressed on the surface of the lymphocytes and their corresponding ligands that are expressed on the endothelial cells lining of the vascular vessel wall. Lymphocyte rolling, that is the contact between the lymphocyte and the endothelium, is initiated by the lymphocyte, that is mediated by repeated binding or the release of the events between selectins and their ligands. During the process of rolling, chemokines are displayed on the surface of the endothelial cells that can interact with the chemokine receptors on the surface of the lymphocytes. Signaling simultaneously interface between a chemokine and its receptor results in a cascade of intracellular signaling that may further activate the adhesion molecules of the integrin family to translate into signals. This happens with the G protein pathway. Firm adhesion, that is, once the integrins are activated, the lymphocytes display high affinity binding sites that interact with the cell adhesion molecules on the vascular endothelial wall, resulting in slowing of the lymphocytes, its arrest and strong adhesion, a process similar to the leukocyte adhesion. Transmigration is the fourth step in the cascade of events, which is not very well characterized as compared to the other steps, especially in terms of T lymphocytes. What is interesting is that the tissue specificity of the T lymphocytes helps in the transmigration, as it does not occur indiscriminately, but is highly regulated to enable the T lymphocytes to precisely enter the specific tissue. Since venules in different tissues display distinct combinations of adhesion molecules and chemokines, ligands, only T cells expressing the accurate combination of the chemokine receptors and adhesion molecules will be allowed to enter into the specific tissue. For example, the chemokine CCL25 and the CAM and the mucosal associated CAM1 are involved in the recruitment of T cells that express the chemokine receptor CCR9 and the integrin alpha 4 beta 7 to the small intestinal mucosa. T cell migration is generally mediated by chemokine integrin activation, which directs the chemokines to provide critical guiding signals to direct T cell migration and to specific tissues within the body. Evolving substantial support that activated T cells migrate to certain vascularized tissues without chemokine receptors or with the inhibition of chemokine receptors. The chemokine independent mechanism generally involve LFA1, that is the leukocyte functional antigen 1, through which the T cells further activate integrins, induce adhesion and subsequent cell migration. The control of T lymphocyte motility is fundamental to the multi-step method of extravasation from the bloodstream into the tissue. Again, the mechanisms will involve tethering, rolling, firm adhesion, and followed by transendothelial migration, which has already been discussed in detail. Remarkably, on interaction with endothelial cells, T lymphocytes crawl with an amoeboid mobility and motility. The shear stress exerted by blood flow contributes to the creation of LFA1 dependent adhesions. The migration of T lymphocytes is oriented against the blood flow as they interact with the innermost surface of the blood vessels. The mechanotaxis of upstream flow relies heavily on passive self-steering mechanisms. Lymphocytes exhibit dynamic protrusions during the transendothelial migration. These actin-rich exploratory structures are regarded as podosomes or philopodia, depending on the state of activation of the lymphocyte. Apparently, they allow the lymphocytes to scan the endothelial cells and to ascertain areas that may be favorable for transcellular diapedesis. The microenvironment of the endothelium also determines the various requirements of the T lymphocytes. This is clearly evident by the role of uropod contractility in transendothelial migration to access the lymph nodes, but does not reach the bone marrow. Now, we go to the third part, that is the monocyte trafficking. Monocytes play a fundamental role in tissue homeostasis, in the defense immunity, and both enhance and reduce the inflammation. Several of the functions of monocytes are carried outside the vascular compartment, hence migration and trafficking are extremely necessary. Recruitment of blood monocytes to the site of injury or infection and their diapedesis through the endothelium, which is again called as extravasation, are critical events at the onset of inflammation, followed by differentiation and successive downstream signaling or cascade of the inflammatory response. Again, a tightly regulated multi-step process consisting of a series of interactions between the endothelial cells and the cells of the immune system precedes the actual transmigration step. For monocytes, this progression is basically believed to track the cascade paradigm which has been initially described for the neutrophils. That means there is a transient activation of the endothelium which is initiated by the inflammatory cytokines such as tumor necrosis factor or interleukin 1b that originates from the tissue macrophages. They elicit a quick expression of additional molecules such as E and P selectin, the ICAM, that is the intracellular adhesion molecule, and the VCAM, that is the vascular cell adhesion molecule, and the presentation of the bound chemokines on the luminal surface of the endothelium. The selectins interact with the O-glycosylated carbohydrate ligands that are displayed on the P-selectin glycoprotein ligand, that is the PSGL-1, and are constitutively expressed on all monocytes. This interface allows the monocytes to roll slowly on the endothelium and balance out the high shear stress which is applied by the flow of the blood vessels. PSGL-1 expression is significantly higher in the inflammatory tissue, which is expressed by IL-6C HI monocytes than by the residents which is expressed as LY6CL, that is the low monocyte, that facilitates their adhesion to the arteriosclerotic lesions. That is, even in arteriosclerosis, which is characterized by an upregulation of the endothelial adhesion molecules like E and P selectin and VCAM in the lesion areas. So, that is deficiency in P and E selectin as well as ICAM has significantly shown to reduce arteriosclerotic lesions in the experimental mice individually. That means, in the conditions of inflammatory response, monocyte rolling will largely depend on the monocyte expressed very late antigen, that is VL4, and the integrins alpha 4 and beta 1 and CD44. CD44 is generally expressed by activated macrophages. The endothelial adhesion molecule VCAM binds to VL4 and facilitates slow rolling on cytokine activated endothelium, thereby ensuring the switch between rolling and firm arrest. The firm adhesion of monocytes to the endothelium is synchronized by CC and CXC chemokines. That means they should have a corresponding CCL receptor along with interleukin 8. Monocytes may use VLA4 for firm adhesion. The monocyte arrest is followed by a directional chemotactic and mechanotactic step in which the monocyte again spreads, polarizes, and consequently locomotes alongside the endothelium to the desired sites for the extravasation. The intraluminal crawling depends on the leukocyte integrins, lymphocyte function associated antigen, and macrophage 1 antigen, that is called as MAC1, as well as the ICAM1 and ICAM2. By blocking these adhesion molecules, the crawling and the subsequent transmigration has been experimentally restricted. This step involves probing the apical surface of the endothelium with the monocyte membrane protrusions, which are called as lamellopodia. Now again, it is important to understand that this crawling is markedly distinct from petroling, which is definitely intravascular and does not lead to extravasation in a steady state. Finally, the monocytes have to negotiate with the endothelium to exit the blood vessel, the lamina bacillis, and the embedded pericytes. Transmigration seems to be the critical event in the monocyte adhesion cascade and is the only step that is never reversed. There are two phases of monocyte transmigration across the vascular endothelium. The thin monolayer of endothelial cells, that is, the cell thickness should be around 0.1 micron in the periphery to 1 millimeter over the nucleus, which constitutes the primary physical barrier between the blood and the tissue. Endothelial cells have no true tight junctions, adherence junctions between the cells, and integrin and cadagrin anchors to the basement membrane that form a network that tightly regulates vascular homeostasis, restrains the leukocyte transendothelial migration. So, three types of migration has been seen in monocytes, as the monocytes prefer transcellular migration in only 10 to 30% of the events. There are strong activating stimuli that increase the overall occurrence of the transcellular transendothelial migration in monocytes. Live cell visualization in vitro and live imaging through confocal intravital microscopy have greatly advanced our understanding in this field. The predominant paracellular route that leads monocytes through the junctions between endothelial cells and requires junctional remodeling, that is a different route directly through fusing of the vesicles in the endothelial cell cytoplasm, constitutes an established mode of monocyte transmigration. Over the past 20 years, it has become increasingly evident that the innate immune system has a central role in polarizing the adaptive immune response towards T helper cell, Th1 or Th2 cell, or to humoral responses. Antigen presenting cells determine the specific recruitment and positioning of the T and B cells in the body. We are improving our knowledge of the recruitment of monocytes from the blood to inflammatory tissues and the capacity of monocytes to differentiate into dendritic cells. For example, new molecules such as RAP1, its ligand PYK2, and atypical PKC have been added to the chemokine induced signal transduction cascade that leads to integrin activation and leukocyte migration. Another important discovery has been the involvement of protein complexes in the establishment of cell polarity, which is again crucial for the directed migration of leukocytes. Furthermore, new adhesion molecules such as JAM and CD99 seem to be involved in the processes that occur at the vascular junctions and function as counter receptors in leukocyte adhesion. Recently described monocyte subpopulations also contribute differently to mount an efficient specific immune response. However, despite considerable progress, several questions yet remain to be answered. Does the constitutive migration of monocytes contribute to the establishment of peripheral dendritic cells? Is a danger signal required for this kind of recruitment? Is the level of integrin activation crucial for the differentiation and migration of monocytes? What are the microenvironmental factors that control the differentiation of monocytes into dendritic cells or into macrophages? Answering these questions will help us to understand the plasticity of the immune system and its impact on the maintenance of immune tolerance. Thank you.